Hello students, so today we are going to learn the fourth part of the chapter 4 molecular basis of inheritance. In this part of the chapter we are going to learn genetic code, characteristic of genetic code, that is the definition terms, mutations and genetic code, and translation in protein synthesis. In the earlier part of the video we learned about transcription, that is from DNA to mRNA by transcription process. So in this part we are going to learn the translation process, that is from mRNA by translation to the formation of polypeptide chain, that is the proteins. Now moving further, here I added two animation image, that is which you can see one the DNA structure with the complementary nitrogenous bases in the pool, that is ATGC pattern, and the right side you can say the genetic code which is shown in a movie called Matrix. You might have seen this, how they get transformed with the help of the coding. So just an idea in future how this coding will be a important aspect. Now moving further to genetic code. So the introduction part of it, DNA is a master molecule of a cell that initiates, guides, regulates and controls the process of protein synthesis. Now as we know, DNA being the master molecule which carries out all the activities right from controlling, regulating, guiding the protein synthesis. Further, to perform this complicated function, it must carry the requisite information for the synthesis of proteins. The site for storing this information lies in the sequence of nucleotides, that is the nitrogenous bases, as evident by Yanofsky and Sarabhai in the year 1964. Now this is the reason why in the previous slide I showed you the DNA animation image where the nitrogenous bases forms an important part in the genetic code. Then further, about 20 different types of amino acids are involved in the process of synthesis of proteins. DNA molecule has 4 types of nitrogenous base to identify these 20 types of amino acids. So basically there are 20 types of amino acids which you are going to learn further, but in combination there are in all 64 types. According to FSA Crick, this information is stored in the form of coded language called genetic code. That's why in the previous animation image, second image, as you saw the Matrix movie, how the genetic code can be crucial in future aspect. That contains code words or codons, each one representing specific amino acid. Genetic code is a collection of base sequence that corresponds to each amino acid. A single nitrogenous base in a codon will encode for only 4 different types of amino acid. A combination of 2 nitrogenous base or doublet codon will specify only 16 different types of amino acid, and a combination of 3 amino acid or 3 nitrogenous bases triplet codon will specify 64 types of amino acids. So as mentioned earlier, now in case of 4 different types of amino acids or in codon, if it is a doublet, then only 16 different types of amino acids will be formed. But if it is in a combination of 3 instead of 4 different types, then it will be a triplet codon, and thereby we can have 64 different types of amino acids. Now here we can see all the 64 types of amino acid. As I told, even though in pure form there are 20 amino acid, but as combination of different sequences, here we have 64 types. But if you see for example vertically and horizontally UCAG, when the combination takes place, you can see all the 20 different types, and certain codes are specific for specific amino acid, and there are certain codes which may be 2 or 3 in number which may code for same amino acid. For example, you can see here UUC, which is for phenylalanine, whereas UUA, UUG, CUU, CUC, CUA, CUG, all codes for leucine. Similarly, the different codons are present. So in all there will be 20 types, but with a different combination 64 you can see, and mainly you can see AUG, that is methionine starting codon, whereas UAA, UAG and UGA, all three are the stop codon. Okay, so you can see a dictionary of genetic code, all coded in the triplet form. Now, coming to the scientist, Dr. Hargobind Khorana. He devised a technique for artificially synthesizing mRNA with repeated sequence of known nucleotides by using synthetic DNA. Dr. Khorana prepared chains of polyribonucleotides with known repeated sequence of 2 or 3 nucleotides. Example, CUC, UCU, CUC, UCU. This resulted in the formation of polypeptide chain having two different amino acids placed alternately, that is leucine and serine. Similarly, polynucleated chain with three nitrogenous bases repeats gave polypeptide chain with only one amino acid. Example, CUA, 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 that is leucine. Okay. Now, a little bit in detail about the scientist. Hargobind Khorana was an Indian American biochemist born on 9th January 1922 and died on 9th November 2011. He shared the 1968 Nobel Prize for Physiology of Medicine with Marshall W. Nirenberg. He got many awards such as Lucia Gross Horwitz Prize, Albert Lasker Award for Basic Medical Research, Padma Vibhushan, Canada Gardner International Award, National Medical Science for Biological Sciences. Now, coming to the characteristic of genetic code, there are different definition or terminologies. So, the genetic code of DNA has certain fundamental characteristics. Now, first one, genetic code is a triplet code, which you already saw. It is a triplet code. So, sequence of three consecutive bases constitute codon, which specifies one particular amino acid. Base sequence in a codon is always in the 5' 3' direction. In every living organism, genetic code is a triplet code. So here, as you already saw the amino acid sequence, which is a triplet codon, it is always in the 5' 3' direction, and this genetic code is true or similar for every living organism, and every codon is specific for a particular amino acid. It will not change for different amino acids. For one amino acid there's a particular codon, as you saw earlier. Like for example, starting codon is which? AUG, which is for methionine, and it will remain the same. Further, genetic code has distinct polarity. Genetic code shows definite polarity, that is direction. It is therefore always read in 5' 3' direction and not in 3' 5' direction. Otherwise, the message will change. Example, 5' AUG 3'. So again, as we saw, the triplet codon is specific for specific amino acid, as AUG is for methionine, the starting codon, and always it is read in the 5' 3' direction. In case if you change the order, then even the code will also change, right? Further, genetic code is non-overlapping. Each single base is a part of only one codon. Adjacent codons do not overlap. So there is no overlapping over here. If non-overlapping, then with 6 consecutive bases, only 2 amino acid molecules will be in the chain. So it does not overlap, and as mentioned, 6 consecutive bases, only 2 amino acid molecule will be in the chain. Further, fourth one, genetic code is commaless. From the name itself you can understand there is no gap or punctuation mark between successive codons. So if you seen the chart itself, there is no comma. It is a codon which is triplet in each of the cases, or there is no punctuation mark also in between the successive codon. Now, fifth one, genetic code has degeneracy. That is, usually single amino acid is encoded by single codon. However, some amino acids are encoded by more than one codons. Example, cysteine has two codons while isoleucine has three codons. This is called degeneracy of the code. Degeneracy of the code is explained by Wobble hypothesis. Here, the first two bases in their different codons are identical, but the third one varies. So here you come across an important terminology called as Wobble hypothesis, where as you saw there is a triplet codon, but in certain cases the first two codes will be similar, the third one may change or vary. Or as I mentioned, yes, the amino acids are specific, right? Triplet codon, but certain codons there may be two codons for the same amino acid, or in case of three codons as we saw for cysteine, isoleucine, for even for other amino acids. Further, genetic code is universal, means throughout the world it will remain the same. By and large, in all living organisms, the specific codon specifies same amino acid. Example, codon AUG always specifies amino acid methionine in all organisms from bacteria up to humans. That means what? The codon AUG, which as I mentioned is the starting codon, and it is nothing but the methionine. So methionine, that is AUG, will remain same throughout the world, universal, and in all the organism, whether it is bacteria, the lowermost species, or the highest, the human. So from bacteria to humans, it will remain the same. Further, genetic code is non-ambiguous. Specific amino acid is encoded by a particular codon. Alternatively, two different amino acids will never be encoded by the same codon. So, the amino acids will never be encoded by the same codon as we have seen. So, two amino acids will never be having a same codon. It will be different for individual amino acids. Then initiation codon and termination codon. AUG is always initiation, that as mentioned starting codon. In any and every mRNA, AUG codes for amino acid methionine. Out of 64 codons, 3 codons UAA, UAG and UGA are the termination codons. Again, as I mentioned in the genetic dictionary, where AUG, which is the starting codon, that is methionine, whereas the stop codon or the termination codon is UAA, UAG and UGA, which is again specific. Now, further, universal. Usually in all organisms, the specific codons specify same amino acid. Again, as we mentioned earlier, universal means in all the organism throughout the world, throughout the universe, the codon is specific for specific amino acid. Further, the 10th one, codon and anticodon. Codon is a part of DNA. Example, AUG is the codon. As we know, it represents methionine. It is always represented by 5' AUG 3'. Anticodon is a part of tRNA. It is always represented by 3', 5' direction. It is 3' UAC 5'. So as you know, the anticodon AUG, it is UAC, and it always in the other direction, that is 3', 5' direction. Now here if you see, I had put one more chart which looks like a dartboard. Now all of you might have seen the dartboard, even you might have played with a dart, right? The arrows. Here you can see how this 4 amino acid, the nitrogenous bases, that is AGUC or AUGC pattern, how they form the codes or how the codon can be done, and you can easily remember this, right? For example, if you take A over here and the 4 codon is A, U, C, G, all the 4, and how the combinations are done. So AUG, so you get the methionine starting codon, and same way for all the others. If you come to the later part, you can see UAA, UAG and UGA, all are what? The start codon. So as we also read one term, Wobble hypothesis, the first and second will remain the same, third one may change, right? So we can see that Wobble hypothesis also over here, how the first and second always remain the same, third one may change, but the amino acid will remain the same. Clear? So this chart helps us to understand how the formation of the 64 amino acids takes place very clearly. Coming to mutation and genetic code. So here the initial definition term, mutation is a phenomenon in which sudden change in DNA sequence takes place. It results in the change of genotype along with recombination. Mutation is a raw material for evolution as it also results in variation. Now as we know, mutation is a sudden change, or we can say it takes place spontaneously. Maybe there may be different reasons for mutation, that may be chemical change, radiation, etc. But it is a change which takes place quickly, and when this change takes place, it always changes the genotype. Thereby new recombination takes place. When this new recombination is a raw material for evolution. So as we can see, new species are formed. How this new species form? These are because of mutation. Mutation lead to recombination. Recombination form variation, and the variation forms evolution in new species. Clear? Further, during mutation, possibility of loss, that is deletion or gain, insertion or duplication of a segment of DNA results in alteration in the chromosome. Mutation can also occur due to change in a single base pair of DNA. This is known as point mutation. Example, sickle cell anemia. Now here, as you know, as we just learned in the initial definition term, mutation is sudden change. Now this sudden change can also cause deletion or insertion of new segment of DNA, which again results in alteration. Now usually mutation can be useful, or sometimes it can be harmful as well as. Now if the mutation occurs in a single base pair, it is called as point mutation, and which can lead to sickle cell anemia. Okay. Further, deletion of insertion of base pairs of DNA causes frame shift mutation or deletion mutation. So this deletion mutation or frame shift mutation takes place because of deletion or insertion of base pairs of DNA. So insertion or deletion of one or two base changes the reading frame from the point of insertion or deletion. Insertion or deletion of three or multiple of three base results in insertion or deletion of amino acids, and reading frame remains unaltered. Frame remains unaltered from that point onwards. That is, when there is an insertion or deletion of amino acids, the frame where you read along will be unaltered. So the point from where it begins, the frame remains unaltered from that point onwards. So here you can see, again I have added a small chart where you can see the DNA bases. Here you can see HIS, or you can say CAT, the original DNA code for an amino acid sequence. Then replacement of a single nucleotide. So here you can see incorrect amino acid which may produce a malfunctioning protein. So in case here, if there's an alteration taking place, instead of CAT, here it is CCT. So there is a change in the amino acid sequence where it can be incorrect, and that may lead to malfunctioning of the protein. Or as we just learned, the insertion or deletion, it can lead a point mutation or frame shift mutation, and even cause certain disease or disorder. Further, tRNA, the adapter molecule. Scientists consider that there has to be a mechanism in which tRNA will read the codon and also simultaneously binds with the amino acid, as amino acid does not have any special capacity to read the codon. So tRNA is considered as the adapter molecule. This role of tRNA was understood much later. So here as we use the term adapter molecule, which was not known earlier, later on they understood the term adapter or the tRNA, which is considered as the adapter molecule. Further, cloverleaf structure, two-dimensional of tRNA process, an anticodon loop that has bases complementary to the codon. It is called anticodon. So just now again you learned earlier the codon which is present on the mRNA, whereas the anticodon which is present by the tRNA, the adapter molecule. It shows amino acid acceptor and 3' end having unpaired CCA bases, that is amino acid binding site to which amino acid binds. So as we saw anticodon which binds to the codon, that is the binding site where a particular amino acid binds. For every amino acid there is a specific tRNA. Initiator tRNA is specific for methionine, as again we saw for AUG, the starting codon, we have got the particular anticodon. There are no tRNAs for stop codon. Now, except or otherwise, as you saw, all the codons has got anticodons right from the starting codon, but the stop codons does not have any anticodons, so there are no tRNAs for the stop codons. In the actual structure, the tRNA molecule looks inverted L, three-dimensional structure. So here you can see the image from the text, figure 4.14, tRNA, the adapter molecule. Here you can see anticodon loop, the anticodon, the mRNA where tRNA binds with. So intramolecular base pairing, the amino acid binding site, glutamic acid. Further even you can see more animation images where you can see how the mRNA where the codons are present and the anticodon, that is the adapter molecule, how it connects with, which helps in the formation of the polypeptide chain. And here as we just read the inverted L, so you can see here how this adapter molecule looks like a inverted L. Clear? Now moving further into the translation process in the protein synthesis. So up till now or in the previous video we learned about transcription. So now we learn how the translation process takes place. Clear? Now, translation is the mechanism in which codons of mRNA are translated and specific amino acid in a sequence form a polypeptide on ribosomes. All the types of proteins are synthesized by the cell within itself, that is intracellularly. That means the synthesis takes place by the cell in itself, that is intracellular. Further, process of translation requires amino acids, mRNA, tRNA, ribosomes, ATP, magnesium ions, enzymes, elongation, translocation and release factors. So these are the requirement or raw materials for the process of translation. Clear? Further, amino acids form raw material for protein synthesis. About 20 different types of amino acids are known to form proteins. These are available in the cytoplasm. Now again coming to the previous slide where we learned about the dictionary of genetic code, where we saw 20 different type of amino acid and in combination 64 amino acid. So here amino acids which are the raw material which is required for the protein synthesis. Further, DNA controls synthesis of proteins having amino acids in specific sequence. This control is possible through transcription of mRNA. Genetic code is specific for particular amino acid. RNA serves as intermediate molecule between DNA and protein. So two things to learn here. In the second point, DNA controls the protein synthesis where amino acids have a specific sequence. Clear? Further, the RNA serve as an intermediate between the DNA and the protein. As we saw in the central dogma, from the DNA to the protein synthesis, the RNA, which is the intermediate helping in formation of the protein synthesis from the DNA, which takes place from the nucleus, then the mRNA by transcription, and further from mRNA to the polypeptide chains in the cytoplasm of the cell. Clear? Further, ribosomes serve as site for protein synthesis. Each ribosome consists of a large and small subunits. These subunits occur separately in cytoplasm. Only during protein synthesis, these two subunits get associated together due to magnesium ion. So they come together because of these two subunits. A ribosome has one binding site for mRNA and three binding site for rRNA or tRNA. So here it is very clear that there is a one binding site for mRNA and three binding site for the tRNA. And ribosome serves the site for protein synthesis. Now, which are the three binding sites? They are P site, A site and E site. Only first tRNA amino acid complex directly enters the P site of the ribosomes. So further you learn about these three sites as well. So remember, there is one site for mRNA and three sites for tRNA, and these three sites are P site, A site and E site. Where P site, it is directly the tRNA amino acid complex is formed in it. Now moving further, here you can see I added a chart over here for a better understanding of the translation process. So always the images and the animation helps us to provide clear understanding. So in this chart it is very clear which is the mRNA, which you can see here, the strip over here, and which is the codon. So mRNA where codons are present and tRNA where the anticodons are present. So you can see the anticodon, they come and attach or bind towards the mRNA. The ribosome, which is the site of protein synthesis, just now as we read. And also if you see here the structure, which is the smaller unit subunit and the larger subunit, okay, for protein synthesis. So you can see here the amino acids, the peptide bond formation. Now you can see here when the codon and the anticodon forms, how the protein synthesis takes place or the polypeptide chains are formed. Clear? So this is a simple image where you can understand how the chains are formed. That is, you can see the coloration of the beads, right? So you can imagine that each and every bead is single amino acid which gets connected to each other to form the polypeptide chain or the protein synthesis taking place. Clear? Here further you can see the animation of what you just saw the image. Okay, firstly you see the figure from the textbook 4.15 translation protein synthesis. So the three processes are there: initiation, elongation, termination. So you can see A, the initiation. So again you see the labeled part first, so your understanding becomes easier from what you saw in the previous image. Smaller subunit of ribosome. So you can see here smaller subunit and larger subunit coming and getting attached. Clear? Then initiator tRNA, see here in the animation now how it is coming and joining, that is UAC joining to AUG, that is methionine. Then the larger subunit, as you can see, getting connected with the smaller subunit of the ribosome. And most important, the three sites, that is E site, P side and A side, just what you read in the previous slide. Clear? Then B, which is the elongation. From the name elongation, how it continues. So you can see here the continuation process, polypeptide chain link is formed. Again E side and A side and P side where it gets connected with, which you can see here clearly. Then C, termination, where it stops. So polypeptide chain which is formed. Here you can see how the last tRNA which is terminated. So here again we can see the first one, as I mentioned, smaller subunit, the tRNA which binds with the mRNA, codon and anticodon, the larger subunit. And in the second animation, we can very clearly see how one by one, starting from methionine and other amino acids join to form a polypeptide chain. Clear? So this image and animation, both from the slide number 12 and 13, makes our concept very clear. Now, coming to the step-by-step process, mechanism of translation, that is synthesis of polypeptide chain. It involves three steps: initiation, elongation, termination. So first one, initiation of polypeptide chain. Now you first see the image. Again, I put individual part of translation, that is initiation, so you can understand it much more clearer way. So mRNA binding site, so as I showed the small ribosomal unit, here you can see mRNA, the initiator tRNA which comes and binds. So you can see here AUG methionine, where the codons are present, and the anticodons UAC, which is the tRNA, brings the anticodons for binding. So here you can see the P site in the center, where the methionine amino acid binds starts with. Then you can see E site and A site. So translation initiation complex. Clear? Now we can go through the point one by one. First one, of initiation of polypeptide chain, this activation of amino acids is essential before translation initiates, for which ATP is essential. Further, small subunit of ribosome binds to the mRNA at 5' end. Initiator codon AUG, that is the starting codon or initiator codon, is present on mRNA, as you can see here, AUG, right? Which initiates the process of protein synthesis. Initiator tRNA binds with initiation codon AUG by its anticodon UAC. So AUG, where you can see the image, the anticodon is UAC. Clear? So that is why the image explains the concept very clearly. So through hydrogen bonds, it carries activated amino acid methionine in eukaryotes, whereas formyl methyl methionine in prokaryotes. So you can remember this: in eukaryote is methionine, that is the starting codon, whereas in prokaryotes, that is bacteria, it is formyl methionine. Then second point, now the large subunit of ribosome joins with the smaller subunit that requires magnesium ions. Initiator charge tRNA occupies the P site of the ribosome and A site is vacant. So the initiator charge tRNA occupies the P site, that is center one, as we saw in the previous slide as well. So P site, that's why you can see the coloration as well too for the understanding. P site is where the initiator tRNA occupies, and aRNA remains, that is A site remains vacant over here. Moving further to the second step, that is elongation of polypeptide chain. Okay, during this process, activated amino acids are added one by one to the first amino acid. So as you saw, the amino acids getting connected one after the other, or one by one, or one at a time. Amino acid is activated by utilizing energy from ATP molecule. This amino acid binds with amino acid binding site of tRNA. So as we saw, the mRNA which is present, the linear form, whereas tRNA which carries the anticodon binds with the amino acid. This results in the formation of tRNA amino acid complex. Addition of amino acid occurs in three-step cycle. So here a amino acid complex is formed. Again, there are three-step process. First is the codon recognition. That is to recognize the code. That is, amino acid tRNA molecule enters the ribosome at A site. Anticodon binds with the codon by hydrogen bonds. Second, amino acid on the first initiated tRNA at P site and amino acid on tRNA at A site joined by polypeptide or the peptide bond. Here, enzyme ribozyme acts as catalyst, that is to activate the process. At this time, first tRNA at P site is kicked off. So here the catalyst is ribosome, which helps to initiate the process. So amino acid the binds or the first initiated tRNA at the P site. Further, translocation. The tRNA at A site carrying a dipeptide at A site moves to the P site. So initially it is translocated, or first time it is initiated with the P site. This process is called translocation. In translocation, both the subunits of ribosome move along in relation to tRNA and mRNA. Hence, tRNA carrying dipeptide now gets positioned at P site of ribosomes, making A site vacant. At this site, the next charged tRNA molecule carrying amino acid will be received. During this process, first uncharged tRNA is discharged from E site. Now I think you understood the concept. Now as we know, coming to the earlier part, there are three sites, correct? P site, E site and A site, that is in tRNA. The first gets positioned at P site. Now when it gets positioned at P site, then the A site gets vacant. Why? Because the next charged tRNA molecule that is amino acid is received at that site, whereas from the E side the amino acids get discharged. So all the three sites are important. P site for getting position with the amino acid, A site to receive the amino acid for getting next charged tRNA, and E site to discharge the tRNA molecule. Clear the importance of all the three sites? Now, further, this process is repeated as amino acids are added to the polypeptide, thereby forming the polypeptide chain. It takes less than 0.1 second for formation of a polypeptide bond. So you can imagine it takes less than 0.1 second for the formation of the peptide bond. This charged tRNA with its amino acid arrives at A site of ribosome. Anticodon codon bind by hydrogen bond. So both is formed by the hydrogen bond. Polypeptide bond is formed. Second tRNA is discharged from the P side to the E side. So as we saw, in P side is located and from the E side it gets discharged and leaves the ribosome. So the events like arrival of tRNA amino acid complex, formation of peptide bond, ribosomal translocation and removal of previous tRNA, that is from the E side, are all repeated. As ribosome move over the mRNA, all the codons on mRNA are exposed one by one for translation. So every time the ribosomes move over mRNA, the codons of mRNA are exposed, that is for the further translation process to carry out. Clear? Now again I added a chart over here showing the transcription translation in short, where you can see from the DNA forming mRNA, mRNA forming the polypeptide chain. So here you can see very clearly the amino acid of polypeptide chain, the P side which getting activated, the mRNA, the E side and the A side. Okay, then you can see here the energy which is utilized, GTP to GDP. Then here again you can see the A side where it gets activated by the next amino acid getting connected to A side, and then it gets vacant as we just read. And further you can see from here the polypeptide chains are formed, and then in the fourth case you can see the discharged molecules are removed from the E side. Clear? Third, the termination and release of polypeptide. At the end of mRNA, there is a stop codon UAA, UAG, UGA, as we just read earlier. It is exposed at the A site. It is not read and joined by anticodon at any tRNA. The release factor binds to the stop codon, thereby terminating the translation process. The polypeptide is now released in the cytoplasm. Two subunits of ribosomes dissociate and last tRNA is set free in the cytoplasm. mRNA also has some additional sequences that are not translated and are referred as untranslated regions, that is UTR. The UTRs are present at both 5' end before start codon and at 3' end, that is after the stop codon. They are required for efficient translation. So just to understand the concept, there are certain regions present which are not translated. Okay, but these are also important. Why? Because in the beginning and end, both the place, that is 5' and 3' end, these untranslated regions are present, or in short, UTRs. So it can be a question, what is UTR? Untranslated region. So this is required for the translation process to carry smoothly. If I just give an example, now if you have seen the flight planes, the planes, this before there's a runway from the end starting point to end point, so that it can easily take over and land, right? So there's an extended strip at both the ends. Clear? So just you can consider that example here. The translation process, both there are region before 3', 5' and where these are the untranslated region which is required for the translation process to carry out smoothly. Finally, mRNA is also released in the cytoplasm. It gets denatured by nucleases immediately, hence mRNA is short-lived. So a question can be put up, why mRNA is short-lived? Because mRNA is released in the cytoplasm, gets denatured by nucleus immediately after the translation process. Now you can see here I added one more chart over here, or the image over here, starting with the AUG methionine codon. Then you can see here how the protein molecule is formed. So this image shows the new polypeptides being formed, and then in continuous process you can see how it begins with methionine and it continues further, the polypeptide forming polypeptide chain, that is synthesis of protein takes place. Also the starting, the N terminus and the later C terminus. Clear? So now I hope the translation process is also very clear. Now I added few more images for the concept to be much more clear. So you can see again here a image where this blue colored region is the nucleus where transcription takes place, and the other region is the process where translation takes place in the cytoplasm. You can see over here. So this nucleus part where the DNA gets transcripted to mRNA. So you can see here the terms which you learned in the previous video: exon, intron, RNA polymerase, RNA transcript, RNA processing, correct? And then the mRNA from the nucleus moves out through the nuclear pore into the cytoplasm of the cell. So you can see the mRNA. Then here again you got ribosomal subunits. Just now as you saw the shorter subunit and the larger subunit, and then which forms the ribosome, the codon, AUG, and the that is a codon, and the anticodon from the tRNA, that is the adapter molecule. So further you can see amino acyl charged tRNA growing polypeptide chain continuously, the amino acid tRNA which connects in the P side, amino acyl tRNA synthetase. Then you can see the translation process which continues. Okay, then I had another image much more clearer for the concept. First you can come to the lower part. The DNA by transcription forms RNA, RNA to mRNA. So here there is one more breakup. There is before forming mRNA is the RNA which forms the mRNA. mRNA in the nucleus moves out into the cytoplasm, by translation process forms the protein, that is the polypeptide chain, right? Now coming to the image, nucleus where the DNA structure which by the transcription process forms the mRNA. Now you can see how clearly, and through the nuclear pore it comes out. Again, the smaller subunit, larger subunit, which helps in the formation of the translation process. You can see a tRNA, the adapter molecule, getting attached to the mRNA, that is codon and the anticodon. So tRNA bringing amino acid to the ribosome, and here it gets translated, and further mRNA being translated over here. This again you can see ribosome incorporating amino acids in the growing protein chain. So how the chain is starting from the methionine and it continues further. Then tRNA leaves the site and the protein chain or the protein synthesis takes place. Clear? I hope now this concept is very clear of translation as well as transcription from the earlier video. Clear? So the questions based on this particular topic what you have learned today, that is about the genetic code and the translation process. So hope the concept is clear to all about the translation as well. So always go through the image and the animation, which helps to understand the concept more clearly. Okay, so thank you all of you. The one more part of the chapter is there, which I'll be putting up in the 5th video of the chapter. So thank you once again.